A good day to Dr. Huda. I'm Yongming, and today I'm going to talk about the dispersal of marine invertebrate larvae. So, firstly, let's start off with a short introduction where the marine invertebrate larvae is the early planktonic stages of benthic invertebrates, and they are all considered as plankton that may differ from adults, especially in their form, and they will undergo metamorphosis in order to become an adult. So, metamorphosis is the critical transitions for them to shift from free swimming planktonic larvae to benthic juveniles, and it involves major morphological changes. So the larvae stages is very important to the marine larvae due to three reasons. So the first one is because the larvae travels across habitats and this ensures the connectivity and genetic flow among metapopulations. Secondly, is about the recruitment success because as a larvae, it allows them to find suitable settlement for metamorphosis which contributes to the adult population size. And the thirdly is because many larvae can delay their metamorphosis until suitable cues such as sufficient food source and substrate are found, and therefore this increases their survival chances and also known as adaptive competence. Environmental cues, which includes chemical cues and physical cues, also influence largely on the metamorphosis of marine larvae. Examples of chemical cues include microbial biofilms like extracellular polymeric substances, also known as EPS, and quorum sensing signals. Physical cues include substrate texture and stiffness. It is known that pollution can inhibit this process. However, some studies have proved that crude oils and its burnt residues can actually induce premature metamorphosis in these marine larvae. It works by tricking them that the environment is suitable for them to undergo metamorphosis, but it actually doesn't. So this reduces their survival rate. Next, we'll talk about ballast water, which is the water carried in commercial vessels used to trim, draft, and stabilize during voyages. And it is widely known as a method for the transportation of marine larvae through two steps. Firstly is the pickup of water that contains plankton, larvae, eggs, and microbes, and secondly is the discharge of water that contains all of these organisms into a new environment. And the examples of marine larvae that can be transported includes comb jelly that has been proven to cause anchovy fishery collapse, and zebra mussels that clog the infrastructure, and also dinoflagellates that can cause harmful algae blooms. Therefore, this impacts ecologically by altering the food webs, just as the cases of comb jelly as I mentioned before, and it can cause biodiversity loss. Economically, it can cause fisheries collapse, which may burdens the fishermen and also causes infrastructure damage that may increases management costs. Next, we'll talk about biofouling, which is the unwanted accumulation of organisms on submerged surfaces of ships or structures. And there are two types of biofouling, which includes microfouling and macrofouling. These are the five stages of biofouling. Firstly, within minutes, it has conditioning film formation, where there will be attachment of molecules that provides nutrition for the organisms. Within hours of immersions, there will be initial cell adhesion, where weak reversible forces allow microorganisms like bacteria and diatoms to attach. And then within days of immersion, it will shift from reversible to irreversible adhesion and biofilm formation. In this stage, the cell will start to secrete EPS that provides cohesion, protection, and genetic exchange. There will be also quorum sensing in this stage. And within days to weeks, the biofilm starts to mature, which is stable enough to resist environmental stress and predation. At this stage, soft-folding organisms also start to attach. And within months, it will enter the macrofouling stage where it is largely influenced by environmental factors and also ecological relationships. Besides, marine larvae also attracted by chemical cues such as EPS or quorum sensing signals for settlement, and then they will undergo metamorphosis. Later on, they will provide structures and act as a physical cues to attract other folding organisms. In short, marine larvae is very important as they bridges between microbial biofilms and macrofouling communities. However, biofouling has also causes a wide range of impact in marine industry. It has causes an increase in cost directly and indirectly through the maintenance and also efficiency loss from drug and resistance. Examples are in the shipping and oil and gas industries. The same goes to the aquaculture. Besides, on biodiversity and ecosystem, it has causes the species richness and genetic diversity to reduce because the invasive species may modify the food webs and reduce the ecological balance at the native ecosystems. Next, we'll talk about the impact of climate change on marine larvae. So, talking about climate change, we'll think about global warming associated with the rise in temperature. This will accelerate the larvae development, which led to more adaptive metamorphosis. Besides, it will also increases the mortality of the marine larvae if the temperature threshold is exceeded. Besides, it will also allow invasive species to migrate and move to areas where they previously could not survive, for example, waters in the polar regions, and they will threaten the native species. Moreover, there will be a shift in spawning timing, which causes the marine larvae to hatch when there is insufficient food source and substrate availability. Furthermore, ocean acidification can reduce the calcification in benthic calcifiers. In this picture, you can see the shells of an unhealthy organism start to dissolve, and this leads to less protection from predators. Circulation change, which includes outer ocean currents and sea level rise, can connect previously benthic habitats that are geographically isolated, so this will affect the recruitment rate. All of these stresses leaves ecological richness open for the colonization of invasive species that can tolerate a broader range of pH and temperature. Next, we'll talk about how global shipping is linked with the spread of invasive species. So the first one is through the pickup and discharge, which is mentioned before. Secondly, The discharge of water which contains those organisms will cause bioinvasions, especially when the source and recipient ports shares the similar salinity and temperature conditions, as this allows the organisms to survive and adapt quickly. And lastly is the spreading of the organisms from one port to another port through the transfer and carried of commercial vessels. To handle the issue, there are some international regulations that has been established. For example, the International Maritime Organization, also known as IMO, established a ballast water management convention in 2004, where the regulation D1 states that all ships must exchange a minimum of 95% of ballast water at the sea. However, this raises a concern where the tanks cannot be fully emptied and can still carry invasive organisms which later get discharged. Besides, the regulation D2 states a strict limit for the concentration of organisms that can be discharged. However, since ships can discharge thousands of tons of water at once, the amount of individual discharge are still suspected to be too high and can cause invasions. Lastly, the IMO biofouling guidelines in 2011 states some of the preventive measures and cleaning practices such as in-water cleaning (IWC) that can be conducted voluntarily. However, the in-water cleaning can release invasive species into the local waters directly, and the voluntarily base of these measures makes it harder to regulate in terms of maintenance. In terms of effectiveness, the ballast water management is considered to be partially successful because they do not fully eliminate the risk, but biofouling is considered not too effective because of invasive species are still spreading. But some countries like Australia and New Zealand has established their own regional regulations, which is good. In order to improve, we should address smaller organisms, for example, those smaller than 10 micrometers that is previously ignored by the current D2 standards. Besides, we should also treat both water uptake and discharge because the ballast tank is dark and full of nutrients. So even though the organisms are killed when uptake, they can still regrow in the tank and be released into the environment through discharge. For biofouling, it is strongly advised to create a mandatory global treaty and also use alternative coatings. For example, we can mimic shark skins using nanotechnology to reduce the release of toxic chemicals into the water. Next, here are some innovative solutions. For example, the microbubbles implosions, where we can use a system like this to produce microbubbles through a sudden pressure drop. And this can mechanically shear apart and kill those fragile marine larvae, plankton, and also spores. But this method is still in testing, so it requires more monitoring. Besides, there is also a method known as ecological buffering, where they create structures with modified texture and complex surface. However, if poorly designed, they can unintentionally favor invasive species. Last but not least, it is strongly suggested that the marine scientists, governments, and industry should all work together. For instance, the marine scientists should carry out R&D and provide evidence-based recommendations, while the governments should invest in research and also implement binding rules with enforcement. Lastly, the industry should follow strictly the binding rules and also adapt more eco-friendly technologies. That's all for me. Thank you.